Okay, let's just dive right into this explainer on clinical chemistry. Today, we're looking at something that seems, well, incredibly simple on the surface, but is actually the foundational bedrock of modern medicine. We're going to see exactly why a simple little letter sitting next to a number on a lab report isn't just some stylistic formatting choice. It can, quite literally, be a matter of life and death.
To make sure we really nail this topic, we'll follow a quick roadmap. We'll start with the problem with plain numbers, move through the SI system and metric prefixes, look at everyday units in the lab, and wrap up with conversions and patient safety.
All right, section 1, the problem with numbers.
Let's start with what I like to call the dangerous ambiguity of unlabeled data. Imagine for a second you're a doctor. You've got a patient in critical condition. You've rushed their blood work down to the lab, and you are waiting anxiously for the results. Finally, the report comes back, and the only thing printed on the page is this completely naked number, 18.
Think about that for a second. Is 18 dangerously high? Is it critically low? Is it perfectly normal? Without a unit attached to it, this value means absolutely nothing. A number only gives us the numerical value, but it completely fails to describe the dimension of the measurement. Without that dimension, it has zero scientific or clinical meaning. I mean, you can't treat a patient with an 18, right?
Looking back at how things used to be is actually pretty wild. Historically, different labs used all sorts of older metric systems, like the centimeter-gram-second system or the meter-kilogram-second system. It was kind of like the Wild West, and this lack of coordination across different medical disciplines led to massive confusion. Imagine transferring a patient to another hospital, but their labs speak completely different numerical languages. This lack of uniformity directly increased the risk of treatment errors, which ultimately forced the entire medical world to figure out a unified international standard.
Which brings us to Section 2, the SI system explained.
To fix all that dangerous confusion, the world desperately needed an internationally agreed-upon standard. Enter the Système International d'Unité, or simply the SI system. It finally provided that uniform language for scientists and clinicians everywhere. Born from an initiative that kicked off in 1948 and was officially adopted worldwide in 1960, this is now the absolute gold standard for scientific measurement globally. It ensures that a lab result generated in Tokyo is perfectly, seamlessly understood by a clinician sitting in Toronto.
Now, the SI system categorizes units to keep things incredibly organized. And the whole shebang starts right here with these 8 fundamental base units. Think of these as the basic alphabet of clinical measurement. We've got the meter for length, the kilogram for mass, the second for time, the mole for the amount of a substance, and so on. Every single scientific measurement in the known universe is built straight out of this core foundation. But of course, the system doesn't just stop at the basics. We aren't just measuring simple mass or time in a lab. We're measuring highly complex physiological dynamics. So we take those fundamental base units and we mathematically combine them to create what we call derived units. For example, if you need to measure force, you use newtons, which are literally derived from meters, kilograms, and seconds. Need to measure pressure? You use pascals. It's this beautifully logical, modular system that lets us measure wonderfully diverse quantities.
Moving on to Section 3, scaling with metric prefixes.
So, in practical application, clinical values are often incredibly tiny. To avoid having to write out endless strings of zeros for microscopic lab measurements, the SI system includes a brilliant little toolkit of standard prefixes. You divide by 10 to get a deci, by 1,000 for milli, and by 1,000,000 for micro. In clinical chemistry, your absolute best friends are going to be milli, micro, and nano. This is exactly how we precisely measured trace elements in your bloodstream without having to write out 20 decimal places on a chart.
And let's briefly look at the other end of the spectrum to see how this builds upward. We use prefixes like kilo, mega, or giga to multiply those base units for massive quantities. A kilo multiplies by 1,000, a mega by 1,000,000. Now sure, you probably won't be measuring a megagram of a chemical in a tiny patient test tube, but having this standardized scaling system that moves in both directions provides total mathematical flexibility.
All right, Section 4, units in the lab. Let's see how this actually plays out.
Let's look at how these strict SI rules are applied to the specific, everyday working units you're going to encounter on the floor of a clinical chemistry lab. First up, we have the mole. The mole is absolutely essential. It's the SI unit for the actual amount of a substance. Historically, we used to measure things purely by weight, using clunky formats like milligrams per 100 milliliters. But the mole was a real game changer. It shifted clinical measurements away from simple weights to telling us exactly how many molecules of a chemical substance are actually present. you're going to constantly see test results expressed in working subunits like millimoles per liter.
Now let's talk volume. Technically speaking, the official SI base unit for volume is the cubic meter, but practically, a cubic meter is basically the size of a large refrigerator. That is incredibly inconvenient for measuring a tiny blood sample. So, the universally recommended working unit for volume in the laboratory is the liter. It perfectly conforms to the SI system, but it actually makes practical sense for human-scale science.
And of course, since we just talked about prefixes, we scale that working unit down. When you're drawing blood or pipetting reagents, you'll be relying heavily on specific submultiples of the liter. You'll use milliliters for standard fluid draws, and you'll drop all the way down to microliters, nanoliters, or even femtoliters when you're analyzing highly concentrated enzymes or genetic material. A very similar practical adjustment happens with mass. The kilogram is the official SI base unit for mass, but again, a kilogram of a chemical reagent is a huge amount for a lab. So the much more practical everyday working unit for lab technicians is the gram. This allows for beautifully convenient, workable submultiples on your daily lab reports, like milligrams or micrograms.
Finally, Section 5, Conversions and Patient Safety. This is where it really counts.
Now we reach the absolute most critical part of this explainer, how applying these units properly directly impacts patient care. Because the transition to SI units took time globally, and because older textbooks or different regions might still reference historical formats, you have to know how to translate. Understanding how to apply a conversion factor is a completely non-negotiable skill. For instance, if you get a glucose reading in millimoles per liter, you have to multiply it by 18 to understand its value in milligrams per deciliter. Got bilirubin in micromoles per liter? You multiply by 0.058. If you multiply by the wrong factor or divide when you should have multiplied, well, you misdiagnosed the patient. It is literally that simple.
Okay, let's test your active recall with a real multiple-choice scenario pulled straight from the source material. Knowing these prefixes by heart is your very best defense against the medical error. So, quick check. Which prefix represents 10 to the negative 3? Is it micro, nano, milli, or pico? Take a second.
If you said milli, hit the nail on the head, you are absolutely correct. 10 to the negative 3 is a thousandth, which is milli. Just for context, micro would be 10 to the negative 6, nano is 10 to the negative 9, and pico is 10 to the negative 12. Committing these to memory makes reading a lab report totally second nature. When we step back and look at the whole picture, the advantages of this entire SI system are just undeniable. By adopting this logical, modular framework, we allow for the true global understanding of lab results. It dramatically reduces the risk of misinterpretation, it guarantees crystal clear communication among clinicians, scientists, and researchers, and it completely streamlines the conversion process. It really is a universal language of safety.
I really want to highlight this quote directly from the source material. A useful laboratory result is one in which SI units are used. Think about the weight of that statement for a moment. The most perfectly executed chemical analysis, using the most expensive state-of-the-art laboratory equipment in the entire world, is completely useless to a treating physician if the result isn't framed in this precise, standard language.
So I'll leave you with this final thought to ponder. Could a single missing letter, say an M instead of a may, or even a totally blank space next to a number, be the difference between a cure and a catastrophic treatment error? Absolutely it could. In the high-stakes environment of clinical chemistry, the unit isn't just an afterthought. The unit is the context, it's the meaning, and it's the safety net. The unit is just as vital as the patient's data itself. Keep that in mind the next time you're reviewing a lab chart. Thanks so much for joining me on this explainer, and keep learning.